Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics. And we have finished replication, we have finished gene expression, including transcription and translation. So you should feel really good with those. And now we need to look at a way of regulation. Do you remember we talked about in the metabolism chapter that like feedback inhibition and competitive inhibition and how you could regulate enzymes so that you could conserve energy? So that's one type of regulation, and that's based on protein to protein. Remember, like an allosteric molecule bound against the enzyme in the protein and change shapes. But there is another way that organisms regulate their activity, and that is at the DNA level. Just shut the gene off. Don't allow transcription to occur. So that's one type of regulation. Now, prokaryotic cells have a specific form of regulation in that category where you can do that, and that is called an Operon structure. So that's what we're going to do next. We're going to learn at how prokaryotic organisms regulate their gene expression by shutting down transcription or turning it on when they need it. So turning it off and on, okay? And all of this is based on conserving energy, making sure that you don't make stuff you don't need, right? So, so that's what we're talking about next. Okay, so let's go to the PowerPoints. So genetic expression, we're regulating gene expression, and this is called an Operon structure, and it's one way prokaryotic cells do that. Now, most of prokaryotic genes are always on. They're expressed at all times. You need them. You're going to need them all the time. But there are some times that there are genes that are not needed all the time. So this is the types of regulation we're talking about, those types of genes. Why would that happen? Well, there are some pathogens that only make a flagella when they get into our human intestines. Like they go through your digestive tract, through the stomach, and then once they get to the intestines, then they make a flagellum, or something like that, where they only need it at a certain time. So these are ways that we can do that. One way, there's another pathogen, the Pseudomonas aeruginosa, and it comes in and it kind of mimics, it kind of looks like a good guy, a bacteria that's not harmful, and our immune system doesn't attack it. And then all at once, when I have a whole bunch that have already replicated inside a host, they'll start producing toxins. And this is after quorum sensing. You remember when we talked about quorum sensing, and you can tell how many bacteria? It's a way the bacteria can determine the density of the population. So when you have a whole bunch of bacteria and this one knows it, it will turn on a gene and turn on production, and it produces this toxin, which if it had come in producing the toxin, that immune system, that macrophage would have gotten rid of it and it wouldn't be a problem. But if I have a whole bunch of them and they all start producing toxins at one time, they can overwhelm the immune system. So it's a way that the pathogens can get in and kind of hide from the immune system and then do that. So that is an example of a way that they use operons to regulate. So let's learn about an operon and how this works. All right, so there's our gene. This is DNA, and we have a promoter. What happens at the promoter? That's right. This is our signal to start transcription. So this is where RNA polymerase binds to the DNA, and then it will start transcription. It'll continue until there's a terminator. That's not listed here, but it's understood. So this is a promoter to a terminator, and it makes mRNA from there. All right, and then between that promoter and terminator are our genes, and we know that prokaryotic cells can have polygenic regions. So this one actually makes 4 different genes. So this one mRNA strand is going to give rise to 4 different polypeptide sequences that'll form into 4 different proteins. Now, usually when you have polygenic regions, these proteins are related. They're all involved in the same thing. Maybe breaking down lactose. All of these produce enzymes that break down lactose. So usually in this polygenic structure, these genes are related. All right, so that's what you've got. Now in this particular thing, we also have another gene. So somewhere else down the DNA, there's a promoter and a terminator, and there's another gene that's nearby this region, and it's going to make its own mRNA. We refer to this as a regulatory gene. Because it's going to make an mRNA, and then it's going to get translated into a protein, and that protein is going to be involved in regulating this series of genes. So this is referred to as a regulatory gene. It's code for proteins that regulate the operon. How do they do that? They actually bind in this operator region. So there is again, these are just series of nucleotides, but it's a specific order that the protein made from this regulatory gene comes over here to the operator and binds, and it can either turn it on or turn it off, depending on what type of operon. All right, so those are the structures for that, this operator region. I think it referred to as like the traffic light. It's going to be able to turn it off or turn it on by controlling the movement of RNA polymerase. So that's something there. Okay, so let's keep going. There are two types of operons. We can classify operons as an inducible operon or a repressible operon. Inducible operons are usually off. They're not usually able to be transcribed. But if the conditions are favorable, the cell can turn it on and make it and allow transcription to occur. So an inducible operon is usually off, not transcribing, but you can induce it to come on. That contrasts with the other category, repressible operons. Repressible operons are usually transcribed. It's something the cell needs most of the time. So its normal configuration is on, so it's transcribed. But if it gets to a point where you don't need it to make any more of those proteins, it can be repressed or turned off. So repressible operons are always transcribed, but they can be deactivated, turned off, repressed by some proteins we call repressors. All right, so we're going to look at two examples for this, and we will look, actually we're going to look at the tryptophan operon first. So we're going to look at a repressible operon, and the example, there's lots of different ones, but the one we're going to look at in detail is called the tryptophan operon. So let's go to that one where it says the TRP operon. Now, tryptophan is an amino acid. If you've heard of tryptophan, this is the one that they always blame at Thanksgiving for making you sleepy, that turkey has a lot of tryptophan. So tryptophan is just an amino acid. It's something that we need. We either get it by consuming it, by taking it in and breaking down other proteins, or some organisms have the ability to synthesize tryptophan, to make this tryptophan, which is good because you need it. So tryptophan can be synthesized, and in a prokaryotic cell, if we're going to make something, we're going to need enzymes, right? So there is a polygenic structure with 5 different genes on one mRNA that's going to make 5 different proteins, which happen to be enzymes, which when they work together can make tryptophan from something else. Okay, so normally, since we need tryptophan, normally in a prokaryotic cell, this is always on. And the RNA polymerase is binding to the promoter, and it's transcribing this mRNA. The mRNA gets translated, and the ribosomes can bind and make these 5 different proteins. Now remember, this is an operon structure, so nearby is going to be another gene with its own promoter and its own terminator, and it's always going to get transcribed and make its own mRNA, and it's always going to get translated, and it's going to produce a protein. Now remember, a regulatory gene and the protein is involved in regulating this nearby operon. The protein that it makes is a repressor protein, but the shape that it's made in is inactive. The shape that it's made in will not fit in this operator region. So this regulatory gene is always transcribed, always translated. Regulatory genes always are. But in this case, in a repressible operon, it makes an inactive repressor. All right, so this is our normal pathway. Normally, this is always getting made. It makes a protein that can't work because of its shape, and it's always making this mRNA, which gets translated into these enzymes, which are capable of producing tryptophan. But say you just ate turkey, or say that for whatever reason, you have a lot of tryptophan. Either they made a lot, or there's a lot in the environment. Either way, this cell has a lot of tryptophan, more than it can use at the time period. Does this cell still want to use the energy to make more of something that it already has a ton of, a stockpile of, a lot of tryptophan? Well, no, we don't want to do something if it's not needed and we have a lot of tryptophan. So let's take a break. Let's not keep making tryptophan because we have so much. How can we do that? Well, let's repress this operon. Let's turn the transcription off for this. All right, so how do we do that? All right, remember we have all this inactive repressor that's been made. And again, this is one protein. It's very easy to make this one protein, even if we're not using it. But the shape change wasn't right to block it. Well, if I have a lot of tryptophan, tryptophan will actually bind to this inactive repressor that was made by this regulatory gene. It'll bind to this inactive repressor, cause a shape change. Remember, when proteins bind, it can change the shape. And now this is the perfect shape to bind to the DNA in the operator region. And when this complex, the tryptophan and the repressor, which has now been activated, binds, then RNA polymerase can bind to the promoter, but it can't move down the mRNA. So transcription is blocked. It is physically blocked. This is a repressible operon. It's normally on, but if the circumstances are correct, or in certain circumstances, we can turn it off. In this case, the circumstances, we've got too much tryptophan. We have extra tryptophan. It's not needed right now, and it's floating around. It'll bind to this inactive repressor that was made by this regulatory gene, changes the shape, and now this binds to the DNA in this operator region and shuts down transcription. Okay, and this will stay that way. Meanwhile, your cell's still making proteins. It uses this tryptophan. Once it starts using up all this tryptophan, it'll actually pull this one off as well. And then that protein changes back into the inactive form, and then we can turn this back on. Okay, so this is the tryptophan operon. It is an example of a repressible operon, and this is how this works. This is kind of tough for students. Sometimes I lose them at this point. Come on, stay with me, don't get lost. But I'll do the next video and we'll look at a different example of an operon structure, and this is an inducible one. So we'll look at that in the next video. Okay, thanks.